Then after getting all this normalization and correcting the systematic errors, what is the final output that we get? So the final output is basically protein IDs, probabilities, the sequence coverage, etc. And for each protein, we get quant values for each experiment and for each peptide as well. And we also can get grouped quantitation values. For instance, for the control group, you can get the mean or the median, and also quantitative error as well as statistical p-values, either using the t-test or the ANOVA if you have multiple groups of samples. So for the generalization, what we can do is, for instance, in this case, for one representative protein, you have the 3 intensity measurements as well as the median value when you group them for the control versus the difference. So there will be many of them. Here, there is no, as you can see, there isn't that much of a difference taking place in this particular protein. And there are some subtle changes taking place. And there will be significant, seems like it's statistically significant in this particular protein. And this one is going in the opposite direction. So likewise, how to best visualize these thousands of quantitative profile at once and then screen out statistically significant changes. So for such cases, what people have thought of is we can use quantitative ratios between the groups and the p-values. So how do we go about it? So I want to mention here, when you think of quantitative ratios, it does not signify the direction. That means, for instance, if a ratio of 2 to 1 can be designated as 2 divided by 1, or 0.5. So the magnitude does not give you an understanding of the direction. So what people have done is they have gone for using log2 ratios to eliminate this problem, because the ratio is independent of the denominator and the numerator location. So when you can get log2 ratios between the grouped abundances, then we can get the statistical metrics of grouped quantitative variation by basically making distribution plots. For instance, in this particular example, the log2 experimental versus the control distribution that is taking place here. And it is not asymmetric, it is kind of almost a normal distribution where you can obtain the mean value, the standard deviation of 4,586 proteins. And then to identify potential or to screen out, what people have worked on is to generate volcano plots, which basically a plot of log2 experimental versus control in the x-axis and minus log 10 p-value. As you could imagine, p-value, lower the p-value, better the statistical confidence. So if you take minus log 10 p-value, then it basically gives you a way to screen out from the top of the table to isolate the ones that are statistically significant. So I want to highlight here.
Then they'll be really big. The really small numbers will be the bigger numbers.
Bigger numbers, exactly.
After you do the negative log 10 transformation.
10, exactly. Yeah. So in this particular plot, I want to highlight 4 different areas. So it is divided vertically in the 0, 0, where they are basically experimental versus the control. 0 means there is basically no change. And towards the left, anything is basically experimental value has a higher value. And anything that is to your right, to your left, the control has a higher value. Towards the right, the experiment has a higher value. So this is basically visualizing it. And then if you place 0.05 onto p-value here, the value comes around 1.3 value. So you can basically have a line to signify quantitative changes that are statistically significant. So in here, all these above this line are basically statistically significant. Then with TMT type approaches, where the precision is very high and even a log2 fold change, which is closer to 0, can have statistically significant change illustrated. Then you might ask the question, then what whether it is biologically relevant or not? So this is a question that it is a debatable question, but in statistical standpoint, just to have a methodology to screen out, here we assumed that 2 sigma plus or minus the mean value would give us a better way to screen out. So here, what we can do is basically isolate the ones that are less than 2 sigma as well as statistically significant. And here in this case, the experimental sample is less compared to the control, and you can isolate the rest of the other ones where experimental is higher compared to the control. However, it does not mean that these are not biologically relevant. It is just the way that we screen out. So this does not strictly mean this is the only way to go about it, because statistically significant means it is statistically significant according to the data. This approach is not, it is actually a strict methodology, but this is not the best way to go about it. But this is a way to go about if you want to screen out only few candidates. So it depends on how you visualize the data. So it raises many questions than answers, of course, but it is the way typically we go about isolating different targets. I would like to, this is the end of the lecture, and I would like to open for any discussions and any questions anybody have in isolating any all these targets, basically.
So we have a question if you go back, Aruna.
Yes. Yes.
So what the blue numbers that you have designated with the dashed lines coming from them? We have a question that asks what they signify.
So they signify basically the mean plus and minus 2 sigma. So these values were generated using the distribution plot, where you have the mean. Over here it is 0.035 obtained, and if you have the standard deviation derived 0.237, this multiplied by 2 plus this one gives you 0.509. So that is 509 over here, and the other is the
and then what is the mean plus or minus 2 sigma? What does that give you from a statistics perspective?
Yes, so statistics perspective. So it is basically, what it means is in the experimental sample, these proteins basically have lower abundance. I'll try to get an example where I have it here.
You can also say it gives you a confidence interval.
Confidence interval, yes, yeah. Exactly.
So what's the 2 sigma, the confidence interval is 95% confidence, is that correct?
The confidence interval values is very actually very strict here. I didn't calculate exactly. It is mean plus or minus yeah, this is actually maybe around 0.98 or something like that.
Oh, it's higher than that.
It's much higher, yeah. Actually, the if you take the confidence intervals, it will be somewhere here in this value. So this is the most strict way of doing it.
Right.
I would say.
And so that's kind of like a way. Some people will use a p-value as a cutoff, and so that's one way that we do very commonly, because that's done a lot in the literature, where you have a p-value cutoff of 0.005, which is why Aruna has that marked. Yeah. And so so that's one way. So then that would mean everything above that yellow dashed line would be significant based on p-value. Yeah. But if you do a confidence interval based on the mean plus or minus 2 standard deviations, it's going to tell you more about the distribution of all of the data. So that's also represented in the distribution plot. And so by doing that, it will let you have your highest confidence changes which are outside that plus or minus 2 sigma. So Sarah asked if if it's usually normally distributed. I think you did mention that, right, Aruna?
Yes. It is not very normal, but it seems like it is normal. But particularly in phosphoproteomics cases, you would see so many over here and very less over here. It all depends on how you visualize the
data. It also
depends
on the biology also.
Biology as well. Exactly.
So if you look at phosphoproteomics data in the context of something where you have DNA damage, you will not have a normal distribution. So I can tell you that without even doing the experiment, because we've seen it plenty of times. And so it depends on the biology of the experiment. The protein levels, though, and that's the type of data that Dr. Wijeron is showing here, the protein levels do often follow a fairly normal distribution. But you could have something that would disrupt that, like perhaps a disruption of the proteasome or something that causes a very large change in protein abundance. And so, but generally, when we look at this, our null hypothesis, we're assuming there's going to be very little change, so that the changes, the experimental versus the control, are going to have very little effect on the overall proteome. That's the typical null hypothesis. So, and it's good when you're trying to find out if this. The next question, Shara asked if this means it's biologically relevant. So biological relevance is very difficult to define. And a change in protein abundance may or may not result in a change in a biological phenomenon or a phenotype of some sort, and that's why it's very difficult to define. This is also true in RNA sequencing, or even in DNA sequencing. If you're looking at an identification of a mutation, you may have a mutation that might change an amino acid in a protein, but whether or not that's biologically significant, that's a very difficult question to answer. And so you can say statistically that you have a change that is significant based on the rest of the population of the data. And that's what we're describing here. But it's good, when we're trying to answer whether or not something is biologically significant within research, really the best tool that you can have to be able to answer that is to kind of be guided by a hypothesis and by scientific method. So if you're going to have a change, I mentioned DNA damage earlier. So then if you have a change in a protein that regulates the DNA damage response, and then you're expecting that as a consequence of that, you're going to have the change in a number of different proteins at the expression level, then that would be how you would form your hypothesis. And then you would do the experiment and then see if that hypothesis is true or not, based on the justifications that you developed in your research project. And so even though we use proteomics and genomics and RNA sequencing as discovery methods, it's really important that we base the experiment still in scientific method, and we have a principle guiding us that's going to give us some idea of what we might expect to see when we do the experiment. The nice thing about these methods is they can bail you out, because if you get a different finding, even if your hypothesis was wrong, you may have some other biology that might lead you in a new direction and may help you to generate a new hypothesis. So that's really what you want to focus on when you do these experiments, just like any other experiment, like in doing a Western blot. So, yeah.